Hello, today I'm going to be discussing a paper from the Geiger lab and the Borhan lab here at MSU. So they worked to design a fluorescent protein with a large Stokes shift being the distance between the excitation and emission maxima, which corresponds to a redshifted emission, and large meaning over 100 nanometers. Some benefits of which include reducing light scattering, increasing signal depth in tissues, and even reducing or eliminating autofluorescence completely. So fluorochromes that are known to have larger Stokes shifts oftentimes undergo intramolecular charge transfer, or ICT. As you can see in figure 1A, there's a schematic of standard fluorescence compared to fluorescence from an altered excited state, which is a type of ICT. And here, instead of emitting from this A star excited state, it is actually converted to an altered excited state before emission, which results in that redshifted larger Stokes shift. In figure 1B, you can see a specific type of ICT known as excited state proton transfer, which the authors focused on. And this is the mechanism for LLSM orange, which is a photoacid. Here, the molecule is excited and its acidity increases by several orders of magnitude. That enables the proximal amino acid to initiate a proton abstraction. And the exact mechanism isn't known. But a hydrogen bond acceptor must be positioned near the phenolic hydroxyl in order to facilitate that ESPT. And the resultant phenolate relaxes through a redshifted emission to give rise to that observed large Stokes shift. So the vast majority of ESPT molecules are photoacids like this. However, there are photobases, they're just much less common, and they work by extracting a proton in their excited states. In previous work, the authors have reported a fluorine-based super photobase known as FR0SB, which you can see in figure 2A here. It's capable of deprotonating either primary or secondary alcohols, and it can exhibit a pKa change of more than 14 units. The protonation results in a highly redshifted excited state, and it has an apparent Stokes shift of more than 250 nanometers. So those findings inspired these authors to incorporate a similar photobase into a rationally engineered protein receptor, in this case CRBP, which is cellular retinol binding protein, to generate a photoactivatable fluorescent protein with a large Stokes shift. So the engineered protein works as shown in figure 2C here by forming an amine with specific aldehydic ligands through a lysine residue in the binding site. And then upon photoexcitation, the ESPT occurs, purportedly through protonation of the amine nitrogen via another nearby binding pocket residue, and that generates the aminium in the excited state. In this case, the authors engineered a glutamic acid into the binding site to serve as that proton donor, and they ended up using these two weak photobases shown in figure 2D, and found they were the most successful out of all that they tried. So like I said, they engineered a glutamic acid to serve as that proton donor, and you can see a schematic with one of those ligands in the binding site here. And so while they don't know the exact mechanism, it's believed that the water molecule highlighted in figure 4B serves to facilitate that proton transfer in the ESPT. And the resulting complex exhibited an apparent Stokes shift of over 200 nanometers and a quantum fluorescence yield of over 70%, which those numbers make it sound very successful. And it's also thought to be the first system of its kind, which is pretty cool. They also demonstrated the implementation of their photobase initiated ESPT system in live cell imaging, and you can see that here in figure 6B.